I intentionally made this talk kind of unstructured, and I just want to give an impression of some of the non-genetic contributions to phenotype. That is, things that can't be deduced from an accurate knowledge of the entire sequence of the genome. So I'll give you some examples of genotype-independent phenotypic variation. And we'll start with something that's probably familiar, and that's a cat. There are several things going on here. First of all, there's probably a piebald mutation that affects melanocyte migration from the neural crest into the skin and the iris. So this eye has been colonized by melanocytes. This one has not. There's also important components, neural crest-derived components in the ear, and this animal is likely to be deaf on the side that shows the lack of coloration in the iris. So this white hair is also due to that melanocyte migration defect. This patchy coloration, this tortoiseshell, is due to X inactivation. So there's genes translocated to the X such that if it's on the inactive X, you get this lighter colored hair. If this gene is inactivated here, but here the gene responsible for eumelanin production is on the active X chromosome. And this is there's a random choice taken around the time of implantation to inactivate one of two X chromosomes, and that decision is irreversible over the life of the individual. It occurs in all eutherian mammals to a lesser extent in marsupials. But notice that. But if you were to sequence DNA from any part of this animal, it would be identical in sequence, even though the phenotypes are markedly different. Now, another way to look at genotype-independent phenotypic variation is interspecies crosses. So here we see a mule, which is the product of a mare, a female horse, and a jack, a male donkey. And these are familiar. They're very, very few in the city, but you've seen pictures of them. Now, the reciprocal cross is much less well known. This is a hinny. It's the product of a genet, which is a female donkey, and a stallion, a male horse. And this animal is dramatically different in virtually all aspects. And those are summarized on this slide. This is from the Quarterly Journal of the British Mule Society. So this animal has the body of a horse, extremities of a donkey, has long ears and a short mane. It's uniform in color. It's nervous independent and will almost always herd with horses given the choice. And has a donkey-like voice and frequently whickers. That's a sound that horses also make. The hinny looks very different. It has the body of a donkey and the extremities of the horse. It has short ears, long mane, frequently dappled as stars and blazes, which you almost never see in mules. It is rather amiable, and given the choice, will always herd with donkeys. And it has an indescribable squeal that's given only when it's frightened. So both of these animals can be males or females. The females have greatly reduced fertility, but they are fertile. But notice that if you were to sequence these two animals, the genome sequences would be identical. And yet the phenotypes are rather dramatically different. Now, there are other examples. You can make the tiger and lion are quite closely related. They have the same chromosome number. And if you cross a male tiger to a female lion, you get an animal that looks like this. This animal is much larger than either parent. It can approach 1,000 pounds in weight. You can see that it has features of a lion. There's the mane and the characteristic head shape. But there's also striped skin that's present on tigers. The reciprocal cross is very different. It's much smaller than either parent. These animals are about 200 pounds, about 1/5 the size of this. And it has more pronounced tiger facial features, actually reduced striping. But these animals, again, if you were to sequence them, you would find that they have exactly the same genome sequences. And you would not be able to predict that they had such pronounced phenotypic differences. Now, here's another example. I'm not sure about this one, but this is probably X inactivation. It could be heterochromia, where eyes are different colors, probably due to cell migration defect. But this eye and this eye have the same genotype, but clearly they have different phenotypes. And here's yet another example. This is a dog that has the merle coat color phenotype, which is caused by transposon insertion into the silver gene. And these transposon insertion alleles frequently have metastable phenotypes where the gene can be off or on. And that's pattern is set up early in development and maintained through life. This is a male dog, so we know that this variegated coat coloration is not due to exon activation. Instead, it's due to transposon insertion in the silver gene. Notice that this eye is dark and this eye is very pale. So if the silver gene is off, that pigment is not produced. If the silver gene is on, pigment is produced. And this is a professional baseball pitcher who has heterochromia. This eye is light, this eye is dark. He's otherwise said to be relatively normal. But again, the same genotype is present in each eye, even though the two eyes are strikingly different in color. Now, there's a lot of information we gain from twin studies. Here's an example of a pair of monozygotic twins, one of which has Beckwith-Wiedemann syndrome and the other does not. And Beckwith-Wiedemann syndrome is a disorder of imprinted gene expression, which I'll talk about in a moment. But this twin is much larger than this one. She has a very large tongue, macroglossia, which causes the tongue to protrude from the mouth. And this twin has an overgrowth of abdominal viscera and a very high predisposition to a number of embryonal tumor types like Wilms tumor, rhabdomyosarcoma, and neuroblastoma. So the symptoms recede with age. Here's the twins at 14 years old. This is the affected twin. You can actually see that the jawbone has enlarged to accommodate the enlarged tongue. This twin is the unaffected twin, but she has Tourette syndrome and mild mental retardation, whereas this twin does not. And this actually, discordance in identical twins is much more common than you would expect. Here's an example of an individual that is hemihypertrophy. So she actually has Beckwith-Wiedemann syndrome on the right side of the midline and is normal on the right side of the midline. You can see the right side of the face is substantially larger than the left side, and the two shoulders are also quite different. And this switches across the midline. So this individual might have an overgrown right arm and shoulder and an overgrown left leg. Once again, these symptoms recede with age because imprinted gene expression is really only found in the embryo fetus and in early postnatal life. Now here's a recent compilation from National Geographic that these all check out. So this gives the rate of discordance in monozygotic versus dizygotic twins. So you'll notice that stature is very strongly genetically determined. Reading disability and a number of other conditions. But notice a lot of these things that show strong, high rates of discordance in identical twins. And Alzheimer's is very relatively late onset, so there's many opportunities for environmental influences. But schizophrenia has an onset in late adolescence or early adulthood, when these twins will have been exposed to very similar environments. So it's difficult to explain this by environmental influences. So the estimates for heritability of schizophrenia range from or rates of discordance for schizophrenia and monozygotic twin pairs ranges from 35 to about 70%. And another striking thing is that the mean age in longevity, mean difference in longevity in pairs of identical twins is about 10 years. So people think that longevity is highly heritable, but it's not really that heritable at all. Now, here we show an extreme example of genotype-independent phenotypic differences. It's not uncommon for a human oocyte to be spontaneously activated, and diploidy is restored either by fusion with a polar body or by endoreduplication. So this is the result of gynogenetic reproduction. So what we see here is a mass of well-differentiated adult tissues. There's hair, bone, teeth, skin, sweat glands, all well differentiated. Some of these have hundreds of teeth, all well differentiated and identifiable as incisors, molars, bicuspids, and so on. But there's nothing approaching. There's no extraembryonic tissues, and as I said before, it's a completely disorganized mass of adult tissues. It also happens that two sperm fertilize an oocyte, which then loses, and then there's loss of the maternal pronucleus, probably with the polar body. So these androgenetic conceptuses develop as a disorganized mass of extraembryonic structures, especially hydrophic chorionic villi. And there's never any sign of embryonic or adult structures. It's just this mass of hydrophic chorionic villi. This was actually a twin pregnancy, and you can see down here the normal twin. So once again, if you were to sequence these, so if this is fertilized by two sperm, if you sequence this, you would conclude that it's a normal human conceptus, and the same over here. So the sequence of these genomes would be completely uninformative as to the genotype. And here's my picture of normal development that occurs with equal contributions of chromosomes from on the maternal and paternal sides. Now, I read a biography of Anne Hutchinson, a religious dissenter, who was exiled from Boston. And if you read this, she brought forth not one, but was most strange to amazement, 30 monstrous births, blah, blah, blah. But this description actually fits a hydatidiform mole, that's the androgenetic conceptus, quite well. And most remarkably, this is from the diary of John Winthrop when he was governor of Massachusetts Bay Colony. So there was a prominent clergyman, John Cotton, who said that this conceptus consisted of several lumps of man seed without any alteration or mixture of anything from the woman. And he was exactly right. This is an androgenetic conceptus that contains only paternal chromosomes. But how he deduced that there was a complete lack of maternal chromosomes is utterly beyond me, because I don't know how he deduced this, but it's one of the most remarkable biological deductions I've seen, at least from the 17th century. Now, imprinting is a special interest of mine. The first imprinted gene was actually discovered here at Columbia by Argyll Stratiatis and Liz Robertson. It was the IGF-2 gene, insulin-like growth factor 2 gene. And here's what we're looking at here are CpG dinucleotides. These are sites of DNA methylation in mammals or in deuterostomes in general. And you'll notice that there's a region here that's methylated in the spermatozoa and completely unmethylated in the oocyte. And if we look down here at another important imprinted gene, you'll notice that this region is unmethylated in the spermatozoa and completely methylated in the oocyte. And after the gametes combine in fertilization, these allele-specific methylation patterns persist for the life of the individual. So if we look down here at maternal chromosome 7, we have if this region is methylated at the 5' end of the KCNQ1 OT1 gene, this untranslated RNA is repressed, and all of these genes are expressed from the maternal chromosome. If this region is unmethylated, then the KCNQ1 gene is expressed, and all these genes are turned off in cis. And we really don't have a molecular mechanism for this. So how expression of this RNA can turn off genes that are hundreds of kilobases away and in the opposite transcriptional orientation is completely mysterious. But it's well established. If you delete this region, or if this region is topically methylated in the paternal chromosome, all these genes come on. Now, right next to this imprinted cluster is the second imprinted cluster. This is the H19 IGF-2 imprinted locus. And you can see the methylation here corresponds to the methylation here. If this region is methylated in the paternal chromosome, and so IGF-2 is expressed in the maternal chromosome, the maternal chromosome expresses H19, but not IGF-2. And there's about 100 genes that are imprinted in this way. And a former postdoc on Deborah Botes recently did a genome-wide survey and concluded it's very unlikely that additional imprinted genes genes that we discovered. So the total number is on the order of 120. Now, I mentioned methylation, DNA methylation, which is really my specialty. I cloned the first DNA methyltransferase when I was a postdoc at MIT, and then after I started my lab at Harvard Med, we collaborated with Rudolf Heinrich's lab at the Whitehead Institute to knock the gene out, and that really revealed the biological functions of DNA methylation. But DNA is methylated to 5 position of cytosines, almost exclusively in CpG dinucleotides, such that both methyl groups are in the major groove. And they make hydrophobic contacts with neighboring bases and increases the melting temperature of the CG dinucleotide by about 7 degrees, highly stabilizing. Now we're going to talk about the general shape of methylation patterns. And what's shown here is the BIC gene, BCL6 interactor and killer gene, transcribed in this direction. Red is methylate. Here's all the CpG sites. Here's all the CpG sites we were able to test. And this is a good illustration of the structure of the genome. There's a large unmethylated domain around the 5' end of the gene, and other sequences are variably methylated. If we look down here at an SVA transposon, which is still active in the human genome, still proliferating in the human genome, we see that it's heavily methylated, and you see no unmethylated strands. And this is a very general picture. So I amused myself recently by calculating that because there's 29 million CpG sites in the genome, the number of possible methylation patterns is 2 to the power of 29 million. And that converts to about 10 to the power of 7.8 million. And that's an enormous number. And you can calculate there's about 2.3 times 10 to the 23rd cells, human cells on the planet at any one time. And you can, what that tells you, because of the great variability in methylation patterns, what that tells you is that no 2 cells in the entire human population on Earth have the same methylation pattern. They couldn't possibly, given the extremely large number of possible methylation patterns. And how much that actually contributes to human phenotypic diversity, except in extreme cases like those that I showed you, is really unclear. We have, I'll end with a new technology that we're developing with the lab in chemical engineering that should make it as easy to determine the entire methylation status of the genome as it is to sequence the genome. So here's a metagene where we're averaging over 14,000 RefSeq genes. And we found that enhancers have very low CpG density, shown in blue, and about half of those CpGs are methylated. So even though it's 50% methylated, there's almost no methylation in enhancers because they have so few CpGs. If we look at the transcriptional start site, we see there's a peak, very high peak in CpG density right at the TSS, very low methylation density. And if we look at internal exons, we find that there's a spike in both CpG density and methyl CpG for reasons that we don't understand at all. This is highly maladaptive because methyl C, when it deaminates, and it does so at a fairly high rate, is converted directly to thymidine. And about 1/3 of all point mutations, both spontaneous and germline, are CDT transition mutations at CpG sites. So you can't explain this by codon bias. So we don't know why methyl CpG densities are so high within exons. For a time, we thought it might be involved in splice site selection, but there's a spike in CpG and methyl CpG at the stop codon, which is not associated with a splice junction. But we've averaged over many genes here, of course, but if you look at any individual gene, it will resemble this. So it'll have an unmethylated transcriptional start site and partial methylation of gene body sequences. And a surprising finding that was made by John Edwards, a former postdoc in the lab. So here we're looking only at promoters, from about +1 to about -500. And there's 2 classes of promoters, very CpG poor promoters and CpG rich promoters. But regardless of the CpG density within a promoter, it always has about 0.3 methylated sites per 100 kb. So methyl CpG density is largely independent of CpG density. And people are, if you've heard anything at all about DNA methylation, it's that it controls gene expression during development. Well, it doesn't do anything of the sort. You can demethylated cells have gene expression profiles very similar to those of normally methylated cells. Instead, what it does is it enforces monoallelic expression of imprinted genes. It suppresses transposons, and it's key in X chromosome inactivation. So if we look at adult brain or human mammary epithelial cells, we find the methyl CpG densities are extremely similar, both here and in this plot over here. I'll describe the artifact that made people believe that DNA methylation controls gene expression during development a little later. And I think I'll skip this. Yeah, so here's some genome-wide methylation data looking at the beta globin genes in adult mammary tissue where these genes are not expressed at all. And what you'll notice is that some of them are methylated, but most of them are unmethylated, even though they're not expressed at all. And this is a pattern you see over and over and over again. So the genes in non-expressing tissues are largely unmethylated. And here's a study from Tomohiro Kono's lab, where he used our DNMT3L mouse mutant. This mutant fails to establish methylation in growing oocytes. It fails to methylate transposons and imprinting control regions. And when Tomohiro did gene expression analysis, he found that the expression patterns were virtually identical. Here we're looking at the methylation in the mutant versus the wild type. Very little methylation here, but the expression is largely unaffected. You can see a very large correlation coefficient. And there's numerous other lines of evidence that indicate that DNA methylation does not turn genes on and off during development, as is commonly assumed. And here's what we think is going on. So we look at the methylation pattern of the nanogene and the OX4 gene, both of which are pluripotency factors that are expressed only in embryonic stem cells and a few other stem cell types. And it's firmly believed that methylation turns these genes off. But if we look, we find that within a population of non-expressing cells, some of the cells are completely unmethylated across the promoter for both genes. And because none of the cells are expressing either of these genes, whereas some of the cells have completely unmethylated promoters, that tells you that the mechanism of repression cannot be DNA methylation. The logic is very straightforward. So these are CpG poor promoters. You'll notice there's just a few CpGs through this region. And what I think happens is that in the active state, long-term inactivity can lead to partial methylation of CpG poor promoters, and that the binding of transcription factors causes their demethylation, whether by an active or passive mechanism isn't clear. But there's a number of transcription factors that when they bind to DNA, have been shown to induce demethylation of local sequences. The CpG-rich or CpG-island promoters are always unmethylated regardless of whether they're expressed or not. So I think that the expression correlation coefficients, the expression methylation correlations are largely an artifact of this, where there's partial methylation in non-expressing tissues, and activation of expression causes the loss of the sparse methylation. Most genes are in this state. They're unmethylated, whether they're in the inactive or active states. Now, there was a very bad but very influential paper published several years ago that claimed that methylation patterns were very unstable with age. And here we're looking at some whole genome bisulfite data across chromosome 1. And here's the daughter and the mother and the father. And if you look at these in detail, you find that they're virtually identical. And again, if you look closely enough, you can find some age-dependent differences, but they're very, very small. That's the problem with these extremely large data sets. There's a huge multiple hypothesis testing problem. But everything tells us that methylation patterns are established in the germline and transmitted with rather few changes throughout all tissues. And here's some data from Joe Ecker's lab looking at mice from fetal stage to 22 months, which is a very old mouse. Green is unmethylated, red is methylated. And you'll notice that the patterns are very, very similar. So that there's very little age-dependent changes in methylation patterns, much less so than was believed before high-quality data on the topic appeared. Now, our real interest has been in the mechanisms that establish and maintain genomic methylation patterns. And there's 3 enzymes involved in that. DNA methyltransferase 1, shown up here, which I mentioned I cloned when I was a postdoc. It has a catalytic domain related to bacterial restriction methyltransferases, and it has a large N-terminal domain that has multiple functions specialized for mammalian cells. DNMT2 looks just like a DNA methyltransferase in every detail of sequence and structure, but Mary Gall, who was a PhD student in my lab, showed that it actually methylates aspartic acid tRNA at cytosine number 38, and that's its only function. DNMT3A and DNMT3B are called de novo methyltransferases. Unlike DNMT1, they have no preference for hemimethylated DNA. So this enzyme does have a strong preference for hemimethylated DNA, which is the product of semi-conservative DNA replication. And this enzyme is largely responsible for the faithful propagation of methylation patterns in dividing cells. And then DNMT3L down here has similarity to 3A and 3B in framework regions, but it lacks all of the motifs, it lacks all of the motifs required for the transmethylation reaction, and instead it's a regulatory factor. And it's expressed in exactly the stages of gametogenesis where genomic methylation patterns are established. This is you can see that this is a beta geo tagged allele of DNMT3L. You can see that it's expressed only in growing oocytes and not in other cells of the ovary. And here's the testis of a late fetal mouse, and you can see the DNMT3L expression is confined to the seminiferous tubules. And if you section this, you find it's expressed only in prospermatogonia. These are cell types that will differentiate into spermatogonia and undergo many hundreds of mitotic divisions before they enter meiosis. And in collaboration with Dave Alice and Jiadong Cheng, we found that DNMT3L specifically binds to histone H3 when it's unmethylated at lysine 4. And there's a very strong negative correlation between nucleosomes containing unmethylated H3K4 and DNA methylation. So a primary determinant of the establishment of methylation patterns in germ cells is the state of histone modifications. Yeah, so there's striking sexual dimorphism in the timing of de novo methylation in male and female germ cells. So in female germ cells, de novo methylation takes place in the growing oocyte, which is arrested in meiosis 1. So the methylation patterns are established shortly before ovulation. On the male side, they're established into prospermatogonia around the time of birth and have to be maintained for hundreds of cell divisions prior to the cells entering meiosis. And it's recently become clear that, that there's a large paternal age effect, and that a number of disorders, autism and schizophrenia are most prominent among them, that are much more common in the offspring of older males as compared to younger males. And this is, we strongly suspect that this is due to errors in maintenance methylation during this long period of these many cycles of mitotic division. Whereas this is just prior to the methylation occurs just prior to ovulation. And it's really striking that there's a large number of C to T transition mutations in spermatogonia, in spermatogonia that give rise to a large fraction of cases of disorders such as Rett syndrome. And I think I'll skip that. Yeah, so we, in collaboration with Dinshaw Patel's lab at Sloan Kettering, we saw the structure of DNMT1. It was a delight to see because we found that when it's bound by unmethylated DNA, the CXXC domain, which is found in a number of proteins and always binds to unmethylated DNA, interposes a stretch of acidic amino acids between the DNA and the active site of the enzyme. And that's a large part of the mechanism that prevents DNMT1 from methylating unmethylated DNA. There's another protein called UHRF1, which cooperates with DNMT1 to ensure that it only methylates heme-methylated sites. There are some unusual circumstances. There's an exception. But we have a pretty good idea of the molecular mechanism that allows methylation patterns that are established in the gamete to be transmitted unchanged throughout the lifetime of the organism. In the case of X chromosome inactivation, there's never any reversion, any reactivation of the inactive X. So it maintains a decision that might have been made 100 years previously with almost 100% accuracy. And this just summarizes a little bit about what we know about de novo and maintenance methylation. So DNMT3L, 3A, and 3B are all required for de novo methylation in both male and female germ cells. We don't know the factors that cause them to differentially methylate imprinting control regions. And maintenance methylation is mediated. So here's a heme-methylated site with methylation on only one strand. UHRF1 interacts with H3K9, trimethylated, and then hands it off to DNMT1, which methylates the unmethylated strand. So deletion of either of these proteins almost completely erases methylation patterns. Now, there's some diseases directly due to abnormal DNA methylation. One of them is ICF syndrome. This is immunodeficiency, centromere instability, facial anomalies. And they have these characteristic heart-shaped faces with wide-set eyes. And there's a severe variable combined into the deficiency that's usually fatal in childhood. But these individuals have mutations in the DNMT3B gene, and they're unable to methylate classical satellite, also called satellites 2 and 3. So this is an ICF patient. This is a control. And there's some abnormal methylation in other regions of the genome. It's most conspicuous at satellites 2 and 3 on chromosomes 1, 9, and 16. Now, Fragile X syndrome, this is a rare disorder. Fragile X syndrome is the most common form of inherited mental retardation. And it's caused by the expansion of CGG repeats in the FMR1 gene. So fewer than 55 repeats, individuals are normal. If they have 55 to 200 repeats, most of them are normal, but they get this region is unmethylated, that this mRNA is toxic, and they get a Fragile X-associated tremor and ataxia syndrome, which is adult onset and progressive. If the full mutation, more than 200 repeats, triggers de novo methylation of the promoter of this gene, and the Fragile X gene, FMR1, is not expressed, and there's a neurodevelopmental syndrome involving large testes, these characteristic facial anomalies, and mild to severe mental retardation. Now, it's important to point out that it's the methylation that causes Fragile X syndrome, because some of the individuals that have this syndrome, and the full expansion greater than 200 repeats, these individuals have this disorder but not Fragile X. So it's the methylation of this gene that causes Fragile X syndrome, not the triple repeat expansion itself, as is causative in most other triple repeat expansion disorders. Now, there's in one region of DNA methyltransferase 1, there are point mutations that cause a dominant adult onset sensory neuropathy, deafness, dementia, and a number of other symptoms that are highly variable from individual to individual. But this is because this is dominant, and because most neurodegenerative diseases are caused by intracellular protein aggregates. Even though the authors attribute the syndrome to methylation defects, it's much more likely that this protein is aggregating in neurons to cause this HSAM type 1E, as it's called. Now, now methylation of a promoter turns the gene off. I showed you that for imprinted genes. It also silences transposons and it silences genes on the inactive X chromosome. But we don't know how methylation silences transcription. Methylation doesn't affect base pairing. But if we look at that this gene, also called ZAC1, we see that it's methylated in sperm, unmethylated in the oocyte, and this black means that it's variably methylated in adult mouse brain tissue. So I showed this slide already. There, so, and I showed a version of this slide as well. But remember, if this region is methylated, H19 is off and IGF-2 is on. If it's unmethylated, H19 is off and H19 is off and IGF-2 is on. So we wanted to know if we could find a factor that could cause biallelic expression of imprinted genes and could activate transposons. So there was a postdoc in Steve Goff's lab named Dan Wolf who died of a chondrosarcoma at the age of 32. But he was studying a protein called TRIM28. And he found that it was essential for the silencing of exogenous retroviruses in mouse cells. And so we had a wonderful collaboration with Dan. Here's TRIM28. It doesn't bind to DNA. It doesn't by itself repress transcription, but it's thought to recruit other factors that repress transcription. And we were very curious to know what those factors might be. I should mention that if you take a gene, a CPG-rich island gene, and methylate the promoter in vitro and then put it into cells, the gene is off indefinitely. So methylation of promoters strongly represses transcription. So we looked at a class of endogenous retroviruses known as IAP, for intracisternal A particles. And we found we had a partial loss of function allele of TRIM28 called CHA2 for reasons too involved to go into. But you'll notice that when we look at IAP DNA, it's resistant to methylation-sensitive restriction into nucleases, both in the wild type and the TRIM28, whereas in the DNMT1 mutant, it's very sensitive to this methylation-sensitive restriction enzyme. MSP1 cleaves the same sequence as HEPA2, but it cleaves regardless of methylation status. So now we're looking at a northern blot where we see that there's the three major classes of IAP transcripts, 1 delta 1, and 2, are highly expressed in the DNMT1 mutant. And they're expressed at very similar levels in the TRIM28 mutant, even though there's no demethylation here, but there's profound demethylation here. So this is the first factor shown to reactivate methylated promoters. And we wanted to know, and here's further example. This is, we're looking at a series of imprinted genes, H19 and IGF-2 I've already described. So the changes in expression are very similar to those that you see in a DNMT1 mutant. It's lost almost all DNA methylation. However, when we did black 6 versus mus musculus castaneus crosses, which have many polymorphisms, we find that the SNRPN gene is expressed only from the maternal allele in wild type, but from both alleles in the CHA2 mutant. And that was true for every imprinted gene that we looked at. We saw full or partial reactivation of every imprinted gene. Again, this is very similar to what you see in a DNMT1 mutant. And here's some bisulfite sequencing data where you can see that the methylation patterns, whereas expression was biallelic, methylation remains monoallelic. So here only the maternal allele of SNRPN is methylated. The paternal allele is completely unmethylated, but both these genes are expressed, as I showed on the previous slide. So now we have now we've identified the factor that's required for the silencing of methylated promoters. If you go to the textbooks, it'll say that the proteins like MBD2 and MECP2 and CAISO are the repressors of methylated promoters. But if you knock those genes out, there's no reactivation of methylated promoters. But it's so well accepted, but completely not at all established. But everybody thinks that these methyl binding domain proteins are responsible for silencing methylated promoters. So we did a screen. We had DNMT1 wild type ES cells and DNMT1 null ES cells. And we figured that if there's a methylation independent silencing complex, such as the one that Dan Wolf and Steve Goff characterized for Maloney-Murine leukemia virus, we reasoned that there should be factors present in methylated cells, associated with TRIM28 in methylated cells, but not in normally methylated cells. So it's a very simple genetic trick that allowed us to pull out some factors. And the factor that we pulled out was OGT. This is O-GLICNAC transferase. It's the only glycosyl transferase that modifies cytoplasmic and nuclear proteins. Of course, there are hundreds of glycosyl transferases that modify membrane proteins. This is the only one that modifies nuclear and cytoplasmic proteins. And here's the structure of OGT. It's encoded by a single gene, has multiple alternatively spliced transcripts, one of which is specific to the mitochondrion. And this is a reversible, this glycnacylation, as it's called, is a reversible, and this enzyme, O-GLICNACase, removes GLICNAC from proteins. And here's a little cartoon of that. So UDP GLICNAC, OGT transfers GLICNAC to serine and threonine residues in proteins to make this glycnacylated protein, and then O-GLICNACase removes them. So we looked to see what proteins were were first of all, selectively associated with TRIM28 in methylated cells, and second, had abnormal glycnacylation. And there's some interesting proteins. So what we see here, going down this is reduced glycnacylation. This is reduction in quantity. And you see OGT on this log scale is down by about 16-fold relative to wild type. And there's a number of other proteins that are known to be involved in gene silencing. This one is of special interest. And we're looking at it right now in collaboration with Art Skolchi at Einstein. This protein, Cullin-7, was initially kind of mysterious because the Cullins are involved in ubiquitination and protein degradation. And there wasn't an immediate link. But when we went to the literature, we found something very interesting. And that is that there's a 3M syndrome, type 3. It's caused by mutations in Cull7. 3M stands for the Miller, McCusick, and Marveau, the authors of the first case report. But it was later shown that these patients have normal methylation in the H19-IGF2 imprinted cluster, but they express H19 from both genes, and they don't express IGF2 at all. And IGF2 is an autocomparative growth factor, which when overexpressed gives rise to Beckwith-Wiedemann syndrome. Remember, that was the giant baby with the abdominal overgrowth. If it's not expressed, the patients are much smaller than normal. And remember, Cull7 is mutated in these patients. So this is fully consistent with the role for Cull7 in repression, or in the monoallelic expression of imprinted genes. There's another disorder called Russell-Silver syndrome. And in about 10% of cases, it's due to a loss of this methylation imprint from the paternal chromosome, such that all the cells express H19 from both maternal and paternal alleles, and they don't express IGF2 at all. In the normal case, H9 is expressed with the paternal allele and IGF2 from the maternal allele. But the similarity between these syndromes is very, very strong. And some clinicians have suggested that many cases of Russell-Silver syndrome are actually 3M syndrome. And in a number of cases where whole exome sequencing was done, mutations in Cull7 were found. So we have, even though Cull7's biochemical activity did not predict any role in any relationship to DNA methylation, the clinical studies strongly suggest that it is. And so here's our hypothesis. We think so there's probably a zinc finger protein. This is, IDK stands for I don't know. This is text change language. But there's a zinc finger protein that binds to methylated promoters, recruits TRIM28, which in turn recruits OGT, which glycnacylates these silencing factors. And what we think happens is that the glycnacylation maintains these factors as active repressors. And in the absence of glycnacylation, they're phosphorylated or under other modifications that reduce their ability to silence methylated promoters. In the case of unmethylated promoters that are silenced by methylation-independent mechanisms, ZFP809 recruits TRIM28, which in turn recruits other silencing factors. And there's no evidence for glycnacylation in the repression of, say, unmethylated Maloney virus. Now, all this is well and good, but we're actually approaching a kind of a scary point. So here we have a mouse mutant strain, a spontaneous mutant in which an IAP transposon is inserted just upstream of the first coding exon of the agouti gene. Now, agouti is involved in makes the melanocytes in the hair shaft make yellow pigmentation so as to produce a subapical yellow band on each hair shaft. The rest of the hair shaft is black. So, but if this LTR is unmethylated, it drives high-level expression of agouti in all tissues, and it makes them. So here, this is a mouse that has this IAP element is methylated in all tissues, and the mouse has a normal brown coat color. In this animal, all the tissues are unmethylated, and it has a bright yellow coat. And here we see a mouse that's variegating because some of the tissues, some of the cells in each tissue, the IAP is methylated, and others it's unmethylated. But if we look at these mice as adults, these mice are genetically identical, virtually genetically identical. This one has a normal brown coat. It's silenced the IAP element in all cells. This mouse is gigantic. It's obese. It's diabetic. It's prone to a number of different tumor types. These mice are the same age and the same genotype, and you would actually conclude that they're different species when you see it in real life. But the difference between these two animals is as little as 3 methyl groups in one endogenous retrovirus. And about 9% of the human genome is made up of LTRs and full-length and variable length retrovirus, in addition to many other transposons. But remember, the difference between these two animals is as little as 3 methyl groups in one tiny region of the genome, and yet it has an enormous effect on phenotype. Now, the bad part, the difficult part in studying DNA methylation arises from the fact that it's very difficult to establish whole genome methylation patterns, and you can't map repeats, among other things. And you need large amounts of starting material because the bisulfite genomic sequencing procedure is very harmful to DNA. It degrades 97% of the DNA. So in collaboration with Jingyu Zhu in the chemical engineering department, we're developing a new method. So Jing makes S-adenosylmethionine derivatives. Instead of delivering a methyl group, deliver a photoactive group to the 5-position of cytosines in unmethylated CpG dinucleotides. Then we irradiate with 350 nanometer light, and that causes oxidation. The chemistry is much more complicated than shown here, but there's a cycloaddition to make a cyclobutane ring here with some photosensitizer attached out here, and it converts this amino group to a keto. So now when you sequence, you'll sequence all the unmethylated CpG sites in the genome will be sequenced as TPG sites. And because this procedure doesn't damage DNA, the expectation is that we can obtain whole genome methylation patterns in very small amounts of DNA, probably down to with the intention of going down to the single cell level. And this is especially important in cancer biology, where there's often profound derangements of methylation patterns in the common carcinomas. And they're highly heterogeneous, so we really need to do single-cell methylation profiling if we're to determine the role of these methylation abnormalities in cancer. We think that they're adaptive, and that these methylation abnormalities actually kill cells. Other people think that they're part of the carcinogenesis. That's a very controversial topic. But anyway, I just wanted to point out that it should be, we should soon be able to determine whole genome methylation profiles at single base resolution as easily as we can sequence DNA. And here are the people that did the work. Matubular is a postdoc who's done very nice work on methylation-dependent silencing. John Edwards, former postdoc now at WashU, who does all our bioinformatics. Deborah Borges did all the DNMT3L work. Dan and Steve I've already discussed. Jikui Song and Dinshaw Patel solved the structure of DNMT1. The chromatin work that we did was in collaboration with Dave Alice and Emily Bernstein. Emily's now Mount Sinai, and Jiadong Chang is our long-term crystallography collaborator. So I tried to give you a general, I didn't mean this to be too coherent. I wanted to provoke you into considering non-genetic factors in mammalian biology. I mean, there's perhaps upon the completion of the human genome, I think there is an undue emphasis on genetic determinism. And I just wanted to point out that there are many other factors that contribute to the phenotype, pardon me, that are largely independent of genotype. And this to me is a great example. These animals are genetically identical, and yet they're phenotypically so different, and their genomes differ probably by the presence or absence of 3 methyl groups out of the 29 million methyl groups in the genome. So I hope this was helpful.